go for.
Hello, everyone.
Thank you.
We're delighted to see you this evening and to welcome you to a new talk. So our meetup for today is about embracing open source software to implement precision medicine. And this talk will be hosted by Guillaume Desachy, head of biometrics at Pierre Fabre. So I will start by introducing R Ladies Paris. We are a local chapter of the global open source community R Ladies Global. R Ladies Paris was founded on 2016, and our goals are to provide a supportive and encouraging network for our users and to promote learning and collaboration in the R community. So R Ladies Paris serves as a vibrant community for our practitioners based in France, and you can follow us on our social networks. If you haven't already, we are very active on Meetup, LinkedIn, YouTube, and Mastodon. So feel free to scan these QR codes, please. The next slide, please. Okay, let me now talk about the R Ladies organizing team. I'm Shaima Bouganmi, a data scientist and business modeler at BVA company in Paris. I'm also an organizer at R Ladies Paris with Mona Belaid, the president of Ladies Paris. Mona is a data consultant for our data, and she's also our Ladies Global team member. Let me now talk about take this opportunity to introduce our next meetup. There will be a face-to-face event on the 29th April at 7 p.m. At the DataCraft offices in Paris on the theme of creating packages in R and the practical development of statistical learning models with tidy models. Nos speakers are Maria Paula Caldas, a data scientist at OCDE, and Julie Aubert, it's INRA research engineer.
So I would like to present our today's invited speaker. So Guillaume Desachy works as Shaima mentioned, as the head of biometrics at Pierre Fabre Group. He also co-lead the European Biomarker Special Interest Group. He teaches a course on precision medicine at NSAE, where he also serves as a board member of the Alumni Association, and as he graduated from the same school. He's also a member for Article 1 NGO promoting equal opportunities for all. I'm sure we're going to learn a lot from him today. So yeah, please, before we start, as I already mentioned, we plan to record, we are already recording this session. If you prefer not to be recorded, please don't hesitate to turn off your video. Also, if you have any questions, don't hesitate to leave them in the chat box. And at the end, before leaving, we'll have a form like just popping up once you leave Zoom. So don't hesitate to fill out the form and to share with us your feedback. Yeah, just a kind reminder that Ladies is dedicated to providing a respectful network and community for everyone. So you have a COC, so please let's keep this meeting a welcoming one for everyone. Thank you, and the screen is yours. Yeah.
Thank you very much. Let me share my screen. Can I do a sound check, and can I check you can see my screen?
Yes.
Yes.
Cool, perfect. Thank you very much, Mona. Thank you very much, Shaima, for inviting me today. It's a great pleasure for me to be here today. It's an honor to be part of an R Ladies chapter. So thank you very much. Before I get started, can I ask you, all of you, a question? And to answer this question, you can type your answer in the chat. Do you have a younger brother or a younger sister? Do you have a niece or a nephew? Do you have a son or a daughter? And if you do, please type yes in the chat. Okay, yes. 4 kids, brother, yes, yes, Cool. So a younger sister. Terrific. So it seems like we've got some things in common in this room. I'm the father of a 5-year-old daughter, and very recently we were on a mission to find a new bike. Now, on that mission to find a new bike, there was one single prerequisite. This bike had to be pink, and that was my daughter's prerequisite. There was nothing else that mattered as long as the bike was going to be pink. So we went on that mission to find a new bike, and we ended up finding a new bike. And as you can see, the bike is indeed pink. Now, the helmet is also pink, and that was another of her prerequisites. I won't get into the story of how we managed to find one and everything, but happy to chat about it later. So we had this new bike in our house, and we're a family of 3. It's our daughter, my wife, and I. So we had this new bike, and we're like, okay, let's give it a test ride. Let's try it out. It seems to be fitting our daughter, but what about us? So my wife gave it a test ride. Obviously, it did not fit. And then I gave it a test ride. Obviously, it did not fit either. Now, you might be saying, okay, but Guillaume, we're not here to talk about your daughter. We're not here to talk about bikes or any of this. What is the common denominator between this and precision medicine? Well, actually, there are some commonalities between this picture and precision medicine, because we know that one size does not fit all for bikes. And if it does not fit all for bikes, it does not fit all for medicines either. And the underlying concept behind this is the concept of precision medicine. The idea of precision medicine is basically to find a treatment which is going to be suited for each patient. And behind the concept of precision medicine, we talk very often about the 5 R's of precision medicine. And what it means is that we need to find the right medicine for the right patient at the right time, using the right dose and using the right route of administration. And what we mean by route of administration is it going to be a pill? Is it going to be an infusion? That's what we mean. So there's this concept of precision medicine, finding the right drug for the right patient. And very often when we talk about precision medicine, we also talk about the concept of biomarkers. And what I like to say is that the term biomarker is actually quite broad, and it can be considered as an umbrella definition. And I promise that's the very last time I will put that many words on the slide, but I think this is quite important. This is the definition of what a biomarker is. And this comes from the NIH in 2016. And what it states is that basically a biomarker is a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or biological responses to an exposure or intervention, including therapeutic interventions. It's a mouthful. The moment we read this, we can see right away that we can fit so many things under the term biomarker. And what it means is that basically we could fit omics data, we could fit genetic data, so DNA data, RNA data. We could also fit blood pressure. We could also fit the new kid on the block, like digital. So in this slide, you can see many, many different kinds of biomarker. It's very diverse kinds of data. So if someone ever tells you, well, I'm working with biomarker data, you can be saying, well, thank you very much, but what are you actually talking about? Are you talking about data which is extremely broad, like DNA sequencing or RNA sequencing? Or are you talking about something which is much smaller, like blood pressure, for example? So a biomarker, under the term biomarker, you can fit a lot of things. Now, what is important is indeed that you can fit a lot of things behind the term biomarker, but what is at least as important is the roles that the biomarkers can play. And the first role that a biomarker can play is that a biomarker can be prognostic. And what we mean by this is that a biomarker is going to be able to predict the evolution of a disease regardless of the treatment being taken. A biomarker is prognostic when it can predict the evolution of a disease regardless of the treatment being taken. Let's imagine we've got two treatments, treatment A, treatment B. Let's imagine we've got two groups of patients, biomarker negative, biomarker positive. For example, the biomarker positive could be patients presenting a genetic mutation, and the biomarker negative patients could be patients not presenting that genetic mutation. Now, let's imagine we're looking at the treatment response in percentage, and let's look at it for treatment A at first. For treatment A, we can see this. We can see that the biomarker positive patients seem to respond better to treatment A compared to the biomarker negative patients. Now, what I was saying is that a biomarker is prognostic if it predicts the evolution of a disease regardless of the treatment being taken. So let's have a look at treatment B. In treatment B, we can see a similar kind of pattern in the sense that the treatment response to treatment B within the biomarker positive patients is higher compared to the treatment response to treatment B in biomarker negative patients. And actually, the difference in treatment response between the positives and the negatives within treatment A or within treatment B is similar. So in this specific setting, we have an example of a prognostic biomarker. Now you could be asking, okay, but why is it of interest? Why would you be interested in a prognostic biomarker? Well, you can be interested in prognostic biomarkers because with prognostic biomarkers, you can identify patients with the biggest unmet need. For example, if we look at the right-hand side, let's imagine treatment B is on the market. Let's imagine we've got these two groups of patients, biomarker negative and biomarker positive patients. We could be saying, well, the biomarker positive patients are pretty well served at the moment because they've got a treatment, the treatment response is good enough. But now the biomarker negative patients, the treatment response and the unmet need is bigger. So we could focus on this group of patients to develop new drugs. So a biomarker can be prognostic. Now, let me go back to my analogy of bikes. Let's imagine that the biomarker is bike size. Either you've got a small bike or a regular bike. Let's imagine that the treatment is the training frequency. Either you do not train, you train 1 hour a month, or you train 4 hours a day. And the goal is to climb Mont Ventoux under an hour and a half. Now, if you don't know what Mont Ventoux is this very iconic mountain at the south of France, which is ridden by the Tour de France riders. And if you make it to the top of Mont Ventoux as the first rider of Tour de France, you are going to be entering this very special cast of riders. But why am I talking about all of this? Well, I'm talking about all of this because I believe that the size of your bike is a prognostic biomarker of climbing Mont Ventoux under an hour and a half. And the reason why is because no matter how hard you train, I bet that your likelihood of making it to the top of Mont Ventoux under an hour and a half, if you've got a small bike, is going to be tiny compared to if you've got a properly sized bike. So a biomarker can be prognostic. Now, the second role that a biomarker can play is that it can be predictive. And what do we mean by this is that a biomarker can be predictive to the response of a specific treatment. A biomarker is predictive when it predicts the response to a specific treatment. Let's imagine we are in a similar kind of setting compared to previously. We still have the two treatments, treatment A, treatment B. We still have two groups of patients, biomarker negative, biomarker positive, and we're still looking at the treatment response in percentage. This is what we see with the treatment A. There is basically no difference in treatment response for biomarker negative versus biomarker positive. And this is what we see with treatment B. So here clearly, we can see that the biomarker allows to predict the response to treatment B. Now, the idea in here is might be more intuitive than for a prognostic biomarker, because we can see right away that if we've got a predictive biomarker, then we can identify the population of better responders. And I like to call this the holy grail. That's what we are after in the pharmaceutical industry, both from an ethical point of view and from a financial point of view. In the sense, from an ethical point of view, we only want to treat patients who are going to be benefiting from this. And from a financial point of view, if we put ourselves in the shoes of, let's say, the French health authority, obviously, we would not want to have biomarker negative patients receive treatment B. So that's what we have when we have a predictive biomarker. A predictive biomarker is a biomarker which is going to be able to predict the response to a specific treatment. Now, you probably know where I'm going to be going next. The goal is still to climb Mont Ventoux under an hour and a half. If you forgot where Mont Ventoux is still this very iconic mountain. The treatment is still the exact same, the training frequency. Either you do not train, you train 1 hour a month, or you train 4 hours a day. And the biomarker now is different. It is the bike model. Either you've got this very shiny road bike, which is very much suited to climb the top, to go to the top of Mont Ventoux, or you've got this mountain bike, which is probably as shiny and as good looking, but probably less suited. Well, I believe that the bike model is a biomarker predictive of the treatment response, because there are going to be some synergies. There are going to be some synergies between the bike model that you're going to be riding, the bike model that you're going to be using, and your training frequency. There is going to be an interaction between the two, helping you to meet your goal, helping you to make it to the top of Mont Ventoux under an hour and a half. So if I wrap this section, what is a biomarker? A biomarker can be many things. It can be genetic data, it can be blood pressure, it can be many, many things, like the new kid on the block I was talking about with the digital biomarkers. And it can play many roles. I talked about prognostic biomarkers, I talked about predictive biomarkers, and there are also many roles that I did not cover today. Now, our goal in the pharmaceutical industry is to develop new drugs. And the more we develop them using precision medicine, the better. And we need tools to be able to develop these new drugs. And as data scientists, as statisticians, for a very, very long time, the tool of choice has been SAS. And when I entered the pharmaceutical industry, when I entered the workforce, it was pretty much either SAS or R, and you basically had to choose your camp. Now, what is very interesting is that nowadays, most of the talents entering the workforce are talented and skilled both in SAS and R, and we can see some true collaboration between the two languages. And what is interesting is that we can see that there are many profiles of our users in the pharmaceutical industry. So there are the usual suspects, like the programmers, the statisticians, the data scientists, the data managers. I call them the usual suspects of our users. But now there are so many other profiles. Let's say the clinicians, let's say the medical doctors. And that's the beauty of R. R being open source, anyone can basically use it. And up until February of this year, I was working for a big pharma called AstraZeneca. And 3 years ago, we started something. We knew we were standing in front of a magnificent landscape. We knew there were some R users at AstraZeneca. Now, we didn't know who they were, we didn't know where they were, and we didn't know what they were using R for. That was 3 questions we basically could not answer. So we're like, okay, maybe we could do something to further this community. So with a few friends of mine at AstraZeneca, we jotted down some ideas. And we're like, okay, what about a Tidy Tuesday? Or what about a weekly blog post? Or even, what about an internal R conference? And that's how we started this community. And the very first initiative we created was something that we branded as AZ Tidy Tuesday. And AZ Tidy Tuesday was basically a playground to hone data viz skills. And if you've heard of Tidy Tuesday, the idea was the exact same. It was to have one topic, one data set, one. That's the exact same idea as Tidy Tuesday. Now, the big difference with Tidy Tuesday is that we built a story around AZ Tidy Tuesday. Every single time we were promoting a data set, we were basically creating a story, crafting a story, and telling a story of the data set around one of the AstraZeneca values, or around one thing happening at AstraZeneca at that moment in time. And AZ Tidy Tuesday was happening the second Tuesday of each month, and we were promoting it on our internal social media. So in here, you can see an excerpt of the post that I had written back then, and the collaborators had 2 weeks to submit entries. And after 2 weeks, we were featuring a contributor. So in here, you can see the featured contributor. This was the very last edition of AZ Tidy Tuesday. There was a total of 25 editions, and celebrate it a little bit. The last edition of Tidy Tuesday was about songs to give it a festive spirit. So that was our very first initiative. Now, when starting this community of our users at AstraZeneca, the one thing that I tried to keep in mind all along was the diversity of learning, because we all like to learn in different ways. Some of us like to watch videos in YouTube, some of us like to read a book, some of us like to be in the classroom. We all like to learn in different ways. And if you think about it, you can think about it according to 2 indices. The first one, you can call it the self-led learning index. And at the top of it is a bit as if you were basically reading a book. And at the bottom of it is a bit like people who are going to be walking you by the hand. People are going to be helping you out. And then the second index, you can call it the synchronicity index. On the left-hand side, you will be able to go back in time. On the right-hand side, you better attend the event, because if you miss it, if you don't attend live, you are going to be missing out. Now, let's go back to AZ Tidy Tuesday. If we put it in these 4 quadrants, AZ Tidy Tuesday is a mix between self-led learning index and learning by, and so opening a book and people helping, walking you by the hand, because when you create your graph, at first, if you want to implement something which is new, you will have to learn by yourself. But what we did is that for every single entry of AZ Tidy Tuesday, we also were doing some code review. So that's why we are saying you don't just learn by yourself, but people are also going to be helping you out. Now, you can also go back in time, because every single entry and every single edition of AZ Tidy Tuesday is now available on an AstraZeneca SharePoint. So people can benefit from what happened in the past. So that's AZ Tidy Tuesday. Now, I was talking about blog posts, and that's what we implemented right after. The idea of this blog post was to demystify a function, to demystify a package. So for example, in here, again, we were using our internal social media, and in here, it was a blog post about pivot longer, which you probably know. And the idea was basically, okay, let me introduce pivot longer, and let me give a few examples. Maybe a few examples that could be related to what we do at AstraZeneca. Then we knew that if we wanted to further the community of our users at AstraZeneca, we could organize an internal conference. And that's what we did. In February 2022, we organized the very first AstraZeneca R conference. And it attracted more than 500 participants connecting from more than 20 countries. So right after this conference, we were like, okay, there seems to be momentum. There seems to be interest in this community of our users at AstraZeneca. But we can't limit ourselves to just a Tidy Tuesday and to just a blog post. So we started some lunch and learns. And the idea is, well, we know everyone is very busy. Let's keep the format short and sweet. a 30-minute time slot, 20-minute presentation, and 10-minute Q&A. And given that we've got offices in Europe, in Asia, and in the US, we were alternating between time zones so that everyone would feel included. Then what we launched is something that we branded as the 10-to-1. The 10-to-1, the idea was to ask the exact same 10 questions to an experienced R user. And again, when you flip through this, you can see the diversity of learning in action, because you can see that experienced R users all learned to use R in very different ways. We launched a monthly newsletter. We were basically recapping on what had happened in the past month and giving a heads up on what was going to be happening in the next. We organized some workshops with POSIT. We got back to the basics of what a community is. In my opinion, a community is all about bringing people together, is all about making new connections. And that was the whole idea of Let's Meet Each Other. It was a very informal get-together, a 30-minute chat, and we were letting the Zoom magic happen. Depending on the number of participants, we were basically creating breakout rooms, and we were letting Zoom do it randomly. We created something that we called the AZR Hot Desk, first on site in Gothenburg, Sweden, and then online. We organized another R conference, an AstraZeneca R conference in 2023. And the new kid on the block is AZR Ladies. And the idea is pretty much the exact same as R Ladies Paris. The idea is to promote the use of R, the use of open source, the use and STEM within women and underrepresented communities. So that's why it means a lot to me to be part of R Ladies Paris today. So what I'm particularly proud of today, when I look at this, is that at the very beginning, we had only 2 initiatives, the one at the top left. But now we're covering pretty well all the ways of learning, either self-led or not self-led, either synchronous or asynchronous. Now, that's what we had implemented. And in early 2022, there was a consultant who was assessing various activities at AstraZeneca, and he reached out to me as I was leading the community of our users at AstraZeneca, and he asked me 3 questions. The first one was, what is the value of this community? The second one was, what is a key performance indicator of this community? And the third question was, what is the return on investment of this community? And at first I was like, okay, I've got no clue. I have no idea how I'm going to be answering these questions. We started this community as a pet project, and then it grew and it was fun and stuff. And we managed to build new connections, but I could not answer these questions at first. But if we focus on the key performance indicator or KPI, at first I was like, okay, could it be like the number of members of our internal social media group? Because at AstraZeneca, we were using an internal social media, and similarly to Facebook, there are groups. So could it be a KPI? Could it be a key performance indicator? And if it is, then we're doing a pretty solid job, because between April 2021, like February 2021 to be exact, when we picked it up, there were barely 200 members in this social media group. And in February 2024, there were 1,700. So if that's a KPI, we're doing a good job. Now, the reality is that's not really a KPI. That's not really a KPI, because you can work as hard as you want. You won't have a direct impact on the number of members of a social media group. That won't be a direct thing. So, okay, so if that's not a KPI, what could be a KPI? Okay, so if that's not the number of members, could it be the number of active members? And I created the exact same graph, and this is what we're looking at. This is the number of active members every single day in this internal social media group. And well, what we can see right away is that it's quite volatile. So what it would mean is that some days we're doing a very solid job and some days we're doing a really crappy job. So I don't think that's a good KPI. But if that's not a good KPI, then what is a good KPI? And then I kept thinking about it, and I believe that this is the true KPI. The number of initiatives led by this community, that's the true KPI, because you can control this. You can control the number of active initiatives. You can start new initiatives. You can stop new initiatives. That's a KPI. That's your key performance indicator, not some indirect kind of things, like a number of members in a social media group. So that was our KPI. Now, there were some other questions. One question was, what is the return on investment of this community? And again, I had no answer to this question at first. And for a very, very long time, I had no answer to this question. And this changed in May of 2023. And like many things, it kind of happened out of the blue. An executive director at AstraZeneca, an executive director in statistics at AstraZeneca, reached out to a colleague of mine and I, and he sent us an email asking us, okay, have you ever used a specific R package which could be of interest to do some clinical trial design? And my colleague answered first saying, well, I've never heard of it. And that was the exact same thing for me. I had never heard of it and never used it. But I knew that I could leverage the community of our users at AstraZeneca. And if you remember, what I was saying is that we had implemented a hot desk, an online hot desk. So I typed a message in this hot desk, and within 40 minutes, we had identified a subject matter expert. Now, I cannot imagine the time it would have taken us to find this subject matter expert had we not had this community, had we not had this online hot desk. It would have been from one email to another email, or like chatting to someone, etc., etc., etc. It would have taken forever. Okay, so we've got a KPI, we've got the return on investment, but what else do we have? Do we have something else? As I was saying, we organized some internal R conference, and in 2023, the leaders of the organizing committee of this internal R conference were Antal and Tomek. And being the organizers, they worked together. So there is a connection between the two. Now let's take another person, another collaborator who was involved in this organizing committee. That's Jasmine. Jasmine has a connection with Antal and with Tomek, thanks to this organizing committee. Now let's take Austin. Austin has a connection to Antal, to Tomek, and to Jasmine. Let's take Yurka. Yurka is connected to the four of them. And let's take Sarah. Sarah is connected to the five of them. And I've only taken 6 of the members of the organizing committee of this internal conference. And in here, with 6 organizers, that's already 15 connections. Now, if we scale this up to the number of members of our internal social media group, we are talking about 1.4 million connections. And 1.4 million is because there are close to 1,700 members in this community of our users at AstraZeneca. Now, don't get me wrong, I'm not saying that these 1.4 million connections are real, but these are potential connections. And as a member of this community, you know that if you type a message in the hot desk, in the online hot desk, you have access to 1,700 like-minded collaborators using R. Let me go back to this. We are still standing in front of this magnificent landscape. AstraZeneca collaborators are still standing in front of this magnificent landscape. Now, the big difference, 3 years later, is that we managed to build connections. We managed to build connections between teams. We managed to build connections between continents, between expertise. And that's the beauty of this community as of today. And I believe that with strong communities, we can develop medicines the way we develop bikes. And if you don't believe in precision medicine, if you think that one size fits all for bikes and for medicines, look at me and tell me that I can ride the bike of my daughter and that I can make it to the top of Mont Ventoux under an hour and a half. Thank you very much. I'm happy to take questions.
Thank you very much, Guillaume. That was amazing talk.
Yes, very interesting.
Very interesting. I love so much how you talk about the community and its advantages. Yeah, so we have some comments in the chat. A very inspiring talk. Don't hesitate if you have any questions, either to unmute yourself or to leave them in the chat. Maybe I will start asking one question. Yeah. So yeah, you talk about the community. Maybe what would be one main advice would you give to other organization or people looking like to build or grow a community from scratch, and how to maintain a community? Yeah.
I think the first thing is think about the sustainability of it. Think whether an initiative you're starting is sustainable in the long run. And what I mean by this is, if we take the example of AZ Tidy Tuesday, AZ Tidy Tuesday, the idea is, okay, people come up with a graph and then we have a featured contributor, right? But we're also doing some code review. And if you want to do a proper job when you do a code review, it's probably going to take you about an hour for every single entry. So if you've got just even like 10 entries, that's 10 hours just to do the code review. And that was the challenge with AZ Tidy Tuesday. Everyone liked it very much. Everyone liked it very much for the challenge it represented, because it challenged everyone to get out of their comfort zone, but at the same time, out of their comfort zone to create the graph, and also because they were getting the code review, but at the same time, it was a challenge to be able to produce all these code reviews. So ask yourself, is it sustainable? Can you sustain it in the long run? Not 1 month, not 2 months. Can you sustain it for 2 years? Will you have the mojo for 2 years? That's one thing. The other thing is always listen to your community. Always listen to what works and what doesn't work. Because, for example, for the hot desk, we started it on site in Gothenburg, Sweden, at the AstraZeneca offices in Gothenburg, Sweden. And the response was not that big. But the moment we put it online with the Teams chat, it worked brilliantly. So listen to the needs of your community. And if something doesn't work, kill it, stop it. And that's fine. Then you'll have more time to start something else, to start a new initiative, another initiative.
Yeah, absolutely. Yeah, I think also it's important to be surrounded by people who also like believe in the community, like today. So you are two managing ladies pairs along our daily work. So I think like the meaning of having a team and to build and maintain this community is important.
Yes.
Yes.
Yeah, absolutely. I could not agree more. Surround yourself with passionate people, and you'll be able to move mountains. Absolutely.
Absolutely. Yeah. So you have a question from Leo. How did you sustain AstraZeneca Tidy Tuesday? Yeah, good question, because like you changed the job, so you are no longer with AstraZeneca.
So this presentation is based on work that I did as an AstraZeneca collaborator. I don't work for AstraZeneca anymore. I work for a French pharmaceutical company called Pierre Fabre. So someone has taken over the lead of the community of our users at AstraZeneca. But to answer the question on how did we sustain in the long run, we managed to sustain it thanks to members of the steering committee of the community, because behind the scenes, not talking about the conferences, but just talking about the community, behind the scenes, it was actually 15 collaborators working on all these initiatives. So that's how we managed to sustain AZITA-ID2Z in the long run, because we had all these members in the steering committee that could help out with, let's say, the code review, or that could help out with writing the story, such things. So that's how we managed to do it. Now, I was also saying, keep listening to your community, because that's one of the reasons we stopped AZITA-ID2Z. The interest was kind of fading away. So we're like, okay, let's just stop it. There's no reason of keeping just for the sake of keeping it. I'm extremely grateful for AZITA-ID2Z because I think it gave a lot of momentum to this community. So that's brilliant. And the beauty of AZITA-ID2Z is because we were tapping on AstraZeneca values and on things happening at AstraZeneca at this moment, at this specific moment, then we could go beyond the network of quantitative collaborators. One AZITA-ID2Z was, for example, on LGBTQ rights at the moment of Pride Month. So at the moment of the featured entry, we went way beyond the quantitative network. We went way beyond data scientists, statisticians, programmers. And this way, we can also build momentum around this community beyond statisticians, programmers, and data scientists.
Yes, amazing. Yes, Chi, you can go along. Go ahead, yeah.
Right. Thank you very much for the very interesting talk. I really enjoyed it. So I'm a statistician and a R programmer. I'm from, well, I don't look like it, but I'm from Norway actually. So I'm following the development of open source activities in pharmaceutical industry. And I have seen a lot of activities from Roche and Novo Nordisk recently. And I'm quite surprised to find so many activities at AstraZeneca. So is there a reason for them not to have so much, promoting their community online so much? And I'm wondering if AstraZeneca is also trying to, ride the wave of from closed source SaaS to open source technology and things like this, because this might affect a lot of young people when they are choosing the companies to join, because universities, they don't teach SaaS, they only teach R. So I'm thinking that maybe in the future there was going to be a culture clash between the two. So I wonder how it's solved at AstraZeneca, for example.
So thank you very much for your questions. Not being an AstraZeneca collaborator anymore, I cannot speak on the behalf of AstraZeneca, and I've never been able to talk on the behalf of AstraZeneca anyway. I'm not representing AstraZeneca. But when it comes to promoting the community, we've tried to promote it as much as possible in conferences, and that's true that we've heard quite a bit of Novo Nordisk, we've heard quite a bit of Roche making huge progress on that front. When it comes to your second question, which is about open source, I would say that our industry the pharmaceutical industry is very much going through a paradigm shift, and that's what I was saying at the beginning of the second part of my talk. I'm fully aligned with what you said, that it's not only a question of, oh, should we use SAS or not, but it's also a question of talents. And my vision on that front is that if we don't make that move, or if we don't accommodate that move, if we don't accommodate multilingual programming, and I'm a strong advocate of multilingual programming, if we don't accommodate multilingual programming in our processes and in our day-to-day jobs, then we're going to go through very, very hard times to find talents, let's say in 5 years, not even 10, 5.
Okay, I hope this answers your question. I think it's time to close the meetup. Thank you very much again, Guillaume, for your participation. Really appreciate your time. Don't hesitate to join us on April 29. So we'll have a in-person meetup in Paris. We found more details about this on our meetup group. Thank you again for your participation, and good evening. Goodbye.
Thank you. Goodbye.
Thank you very much. Have a good one. Take care.
Thank you.
Bye.
Bye.
Bye.